A single leaf in a museum drawer once rewrote an entire branch of science. And the very first discovery we'll explore is something almost nobody guesses. Stay tuned to uncover it. Imagine opening a century-old drawer of tick specimens and finding the exact creature that first spread Lyme disease. Researchers examined ticks collected in the early 1900s from the northeastern United States, sequencing their DNA and discovering the bacterium Borrelia burgdorferi in specimens predating the first human cases. This unexpected timeline showed that the pathogen was silently circulating decades before it was recognized, reshaping epidemiology and prompting new public health strategies. By comparing historic and modern ticks, scientists also traced how climate-driven range expansions altered disease risk, highlighting the value of museum collections as time machines for emerging infections. The finding underscores that even the smallest preserved arthropods can hold clues to major health challenges, urging continued investment in natural history archives.
Discover that the passenger pigeon, once numbering billions before its tragic extinction, left behind a genetic legacy hidden in museum skins. By extracting DNA from specimens collected in the late 1800s, researchers reconstructed partial genomes and uncovered a surprisingly high level of genetic diversity that modern samples had missed. This insight suggests the species collapse was driven more by rapid habitat loss and hunting than by inbreeding, challenging long-standing assumptions about extinction dynamics. The study also demonstrated that even highly degraded DNA can illuminate population history, offering a template for rescuing genetic information from other vanished species. In essence, the dusty drawers of natural history museums became a portal to a lost world, reminding us that the past still speaks through preserved feathers and bones.
Few people know that a fungus collected over 80 years ago in a remote forest yielded a brand new class of antibiotics. The specimen, stored in a fungal herbarium, was revived and cultivated, leading to the isolation of a compound named mysocidin that targets drug-resistant bacteria by a novel mechanism. Early tests show activity against MRSA and VRE, sparking excitement for a potential new line of treatments. This breakthrough illustrates how dormant microbes in museum collections can become treasure troves for modern medicine, especially as the race against antimicrobial resistance intensifies. Researchers now prioritize re-examining archived fungi, hoping that other hidden pharmacological gems await discovery in the same dust-covered shelves.
In 2026, scientists harnessed digitized butterfly collections spanning more than 100 years to chart how species ranges have moved northward with warming temperatures. By georeferencing each specimen's collection location and date, they plotted shifting elevation and latitude trends, revealing that many temperate butterflies have advanced an average of 20 kilometers per decade. This massive, data-driven analysis confirmed predictions of climate-driven distribution changes and provided concrete baselines for conservation planning. Moreover, the study highlighted gaps in historical sampling, prompting museums to fill those voids for future research. The elegant wings of preserved butterflies thus serve not just as art, but as climate barometers chronicling Earth's warming story.
Deep beneath the Arctic permafrost, a 30,000-year-old horse bone, stored in a museum's paleontological collection, yielded DNA that reshaped our understanding of equid evolution. Sequencing revealed that this ancient horse possessed a unique combination of genes linking modern wild horses to extinct lineages previously thought unrelated. The findings suggest multiple interbreeding events across continents, complicating the simple linear model of horse domestication. Additionally, the genetic data pinpointed adaptations to cold environments, offering clues about how species survived past glacial cycles. This discovery underscores how well-preserved fossils, when paired with modern genomics, can rewrite deep-time narratives.
Hidden in the spleens of rodents collected in the 1960s, researchers uncovered a previously unknown arenavirus using metagenomic sequencing. The virus, named Mongolia-like virus, shares genetic traits with pathogens that cause hemorrhagic fevers in humans, raising concerns about its zoonotic potential. By analyzing the archival tissue, scientists could trace the virus's evolutionary history and identify mutations that might enable cross-species transmission. This proactive discovery, made before any human cases appeared, illustrates how museum specimens act as early warning systems for emerging diseases, reinforcing the importance of preserving and revisiting old samples in the fight against future pandemics.
Astonishingly, a pair of salamander skins stored in a European museum for decades turned out to be the first documented hybrid between two distinct species. Genetic analysis showed that DNA from the fire salamander and the alpine newt had intermingled, producing viable offspring in a narrow alpine zone. This hybridization event, previously thought impossible due to reproductive barriers, revealed that climate-induced habitat overlap can foster unexpected gene flow. The discovery reshapes our understanding of amphibian evolution and highlights how museum specimens can capture rare, fleeting biological events that might otherwise vanish without a trace.
From a single drawer of beetles in a natural history museum, taxonomists identified a cryptic species that had been masquerading as a common relative for over a century. Detailed morphological study combined with DNA barcoding revealed subtle differences in elytra patterning and a distinct genetic signature, prompting the description of a new species named after the museum itself. This finding underscores how even well-studied groups can hide unseen diversity, and it emphasizes the role of meticulous curation and modern techniques in unveiling hidden facets of Earth's biodiversity.
Finally, the fragmented genome of the iconic Tasmanian tiger, or Thylacine, was pieced together from a handful of museum skins collected before the species vanished in the 20th century. Researchers used advanced sequencing technologies to recover enough DNA to reconstruct over 90% of the genome, revealing surprising genetic ties to marsupial wolves and shedding light on its unique reproductive biology. While extinction remains speculative, the genome provides a valuable resource for comparative studies and informs conservation strategies for its closest living relatives. The achievement illustrates how museum relics can resurrect genetic knowledge of species long gone.
Hidden in the pages of herbarium sheets dating back to the 1800s lies a detailed chronicle of how invasive plants have marched across continents. By digitizing and georeferencing these pressed specimens, scientists mapped the first recorded appearances of notorious invaders like kudzu and Japanese knotweed, pinpointing introduction pathways and rates of spread. The data revealed that many invasions coincided with railway expansions and trade routes, offering concrete evidence for the role of human movement in ecosystem disruption. This historical perspective equips managers with precise timelines to better predict and mitigate future invasions, proving that pressed plants can be powerful tools for ecological foresight.
We've traveled from ancient DNA hidden in beetle shells to climate fingerprints etched on centuries-old butterflies, each revelation showing how priceless biodiversity collections truly are. If you love seeing how preserved specimens continue to rewrite our understanding of life, hit subscribe so you don't miss the next mind-blowing list we release each week. Which of these discoveries surprised you most? Let me know in the comments below, and if you enjoyed the journey, give this video a like. Thanks for watching, and keep marveling at the wonders of nature.